Okay. Hi, Jishnu. Good evening. Hi, Jishnu. Can you hear me?
Yes, sir.
Yeah. How are you, Jishnu?
Good evening, sir.
Yeah. How are you?
I'm fine, sir.
Preparing well for the exam?
Yes, sir.
Or prepared well?
I'm preparing, sir. Preparing.
Hi, students. Good evening.
Good evening, sir.
Yeah. So, can you all able to see my screen? Hello?
Yes, sir.
Yeah. Good. So, how was the exam? How did you all do it?
Fine, sir.
Fine. Okay, so the question starts from here, right? For biology. Hello?
Yes, sir.
Yeah. So what's the answer? The component of a chromosome that controls heredity is what's the answer?
DNA, sir.
DNA. Okay. All of you written correctly? All of you written correctly?
Yes, sir.
Yeah. Is there any more specific term can be better than this? DNA, apart from DNA, is there any other term? The component of a chromosome that controls heredity.
So genes?
Okay, genes. Fine. Very good. So the next question. The maleness of a child is determined by? Yes, tell me. What's the answer?
Sir, A.
A. Means Y chromosome.
Yes, sir. Y.
Okay. Okay. Fine. In all of the organisms, only Y chromosome. Is it correct for all of the organism? See, in humans, okay, Y chromosome determines the sex. Now my question is,
No, sir. In other organisms, it need not be.
okay, example, give me some example.
In turtles, it depends on the temperature during
Very good.
fertilization.
Very good. What else determines the sex?
Presence of a chromosome, like in what?
Yes. Tell me. Okay, I'll ask you, for example, in honeybee, how the sex is determined? In honeybee, how the sex is determined in a honeybee? How the sex is determined in honeybee? Forgot, huh? See, in honeybee, the process of fertilization. If the egg is fertilized, the chromosome number is duplicated so that diploid condition develops female. But if the egg is unfertilized, that haploid condition results in the development of male. Remember? You remember that?
Yes, sir. Yes, sir.
Okay, fine. In Drosophila,
Sir, it's ulta, like XY is female, XX is male.
in Drosophila, I'm asking about the fruit fly. See, in Drosophila also sex is like humans only. In birds, like chicken only, that's ulta. Okay, in chicken, homogamety and heterogamety condition. The homogamety condition represents male. The heterogamety condition represents female. Okay?
Yes, sir.
See, yeah, here sex determination connected with humans, but at the same time, please ensure that you are aware of the sex determination in all other organisms, especially honeybee, chicken, or the turtles, etc. Okay, birds and turtles. Keep that in mind. Okay. Next question. Alternative form of a gene are called
Sir, alleles.
allele. Okay. What are called then multiples or multiple allele? What
Not
is
sure.
that? Not sure. Okay. See, if you have or if we have more than 2 genes to determine one character. Okay, more than 2 genes to determine one character, that is called what? Multiple allele. Okay, for example, our blood grouping is determined by IA. Okay, IA, IB, and IO. 3 genes. These 2 are dominant gene. This one is recessive gene. Okay, so 3 together determines our blood group. So multiple alleles are like 2 genes or more than 2 genes. It determines one character. They are called what? Multiple alleles. Okay. Example for alleles. Can someone tell me? Example for alleles. Example for alleles. Hello, students. Example for alleles.
Sir, you mean like name of the allele or
Yeah,
character?
where you studied? Where you studied? Give me some examples.
Pea plant.
Pea plant. Okay, very
Uh,
good. One.
uh,
Yeah, if capital R, that is round.
yellow and green, wrinkled
Okay,
and round.
yellow. Very good, very good. All are contrasting. Okay, very good. Alleles. Fine. What is called the loci here? You have loci. What is it? What is loci? Means location, position of genes on a chromosome. Okay, locus means position of genes. If this is the chromosome and this is the locus, here gene 1 is present, gene 2 is present, gene 3 is present. Okay, so that position of genes on a chromosome is called what? Locus. Okay, and what is chromosome? This one. Okay, the condensed part of DNA is chromosome. Okay, hello, students.
Yes, sir.
Yeah, the reason is why I'm telling all these things. If options are given, you should know the meaning of all other options. What is the correct one? Why the others are wrong? That also you should be thorough with. Because maybe in the exam they may ask other question connected with these options. The position of a gene on a chromosome dash. Then you should know with same options, maybe answer is different. Okay, so keep that in mind. Next. This one. How are the 2 strands in a DNA molecule held together? How many of you attended this question? What is the correct answer? How many of you did not understand this question at all? Hello, students? Hello, students. Hello. Hello, students. Are you all listening? Hi, hello. At least if you don't know answer, you please tell me. I'll help you. Say yes or no or something. Hello.
Yes, sir. You're
Okay. Hope nobody knows the answer of this. So listen. DNA is double standard. Okay. You know that?
yes, sir.
DNA is double standard. For example, here sugar will be there, phosphate will be there, phosphate will be there, sugar will be there, like this. So this is called what? The polynucleotide chain. Okay, that is called what? The polynucleotide chain. Like that, here also you have one strand. Okay, here also you have one more strand. Two strands of DNA we have. Sugar and then phosphate. Sugar and phosphate. Okay. What are the different bonds which makes the polynucleotide chain or DNA? First bond, you can see sugar is connected to nitrogenous base. Okay, here you have the nitrogenous base. Sugar is connected to the nitrogenous bases. Like that, the sugar is connected to the nitrogenous bases. Okay, the bond which connects sugar to the nitrogen bond is 1,4, means 1, you can say glycosidic linkage. Okay, the first bond is actually sugar to nitrogenous base. The bond is glycosidic bond. Second, the sugar to phosphate. Okay, sugar, 5-carbon sugar to phosphate group is phosphoester bond. Okay, phosphoester bond. And then here, okay, here, the two strands, the nitrogenous bases are held. Okay, two complementary strands are held together by what bond it is? The answer is means the nitrogenous base, between nitrogenous base and nitrogenous base. It is actually hydrogen bond. Okay, three type of bonds. Sugar to nitrogenous base. Okay, one nucleotide comprises of sugar, nitrogenous base, and then phosphate. So this bond is called what? Glycosidic bond. And this sugar to phosphate is called what? Phosphoester bond. One nitrogenous base with other nitrogenous base of the complementary strand. That is called what? Hydrogen bonds. Okay. What is the answer? All of you written? Let me check. So the answer is hydrogen bond. So be thorough with. There is a box in your material about the DNA structure. So this is the main thing. Okay, the kind of bond which makes the polynucleotide chain, which makes two strands of DNA. At least now you can
Okay.
understand. Okay. Now this is. We'll go to the next section. What is the question number for next question? Yes. Is this the last question? Hello, students? Yes. Tell me the significance of Mendelian laws. Okay, the question is about the significances of Mendelian laws. Means where we apply Mendelian laws. On your own, you please explain. You can take any example. Hello, students. Yes, Ridya, tell me. Let me point out to you. Biological significance of Graham-Mandel's law. So, okay. Jishnu, what did you write for this question? Hello, students. Why no one is interacting? I just want what you have written. In what way you understood the question? Or you can, yes.
Yes, sir. I wrote first of all that because of Mendel's laws, we know how genes are inherited. That it is inherited from both parents, and there are alleles. There are two genes of the same characteristic that is passed on to the offspring. That's one point I wrote. Then just one more point I wrote. It tells us how variation takes place and how variation is brought about in the offsprings.
Okay, very good, very good. See, because, and this question is like very interesting question in the sense here, you cannot get simply any definitions or some in a quotes in the book. You have to understand Mendelian concept. You have to understand the basic principles of inheritance, and from that you have to write it on your own. Okay, so significance in the sense, see what depends upon Graham-Mendelian experiments. What we understood, how the characters, see the title of Mendel's experiments given us the rules of inheritance. Okay, so if we understand the rules of inheritance, the principles of inheritance, it is easy for us to, for example, how dominant characters are inherited, how to select pure line with dominant character. Okay, so if you know this, then in crop variety improvement, you can select your chapter 10 is about, or not chapter 10, in general, I think in class 9, like improvement of food crop varieties, a chapter will be there. It means if you understand Mendelian concepts, if you know the principles of inheritance, you can apply it for improving a crop variety or getting the crop variety which is having only the good dominant characters. Okay. And of course, since you know the meaning of variation, and you can produce many hybrid plants also. Okay, so Grover Mendel's experiments helped to understand how characters are inherited. So with that, you can do any kind of experiments, like get your results applying Mendel principles. Okay, like if you know the formula, you can solve any any solutions. Okay, any problems you can solve. Okay, know the formula, then you can solve any problem. Similarly, like that. Okay, in plant variety improvement or even in animal variety improvement, if you apply Mendelian law, you get good results. Okay, whatever you want, the desired thing you can achieve. That's the importance of this question. Okay, fine. So we'll go to the next question.
So I think the next question is the 30-second question, sir.
20-second,
30-second, 32, sir.
Yeah. Yes. Name the plant Grover Mendel used for his experiment, and what type of progeny was obtained by Mendel in F1 and F2 generations when we crossed tall and short plants. What ratio he obtained in F2 generation? So answer one by one. So what plant he selected?
Pea plant, sir.
Oh, the scientific name? The scientific name? What is the scientific name of pea plant? Hello students, what is the scientific name of pea plant?
Sir, Pisum something, sir. I remember like that.
Pisum sativum.
Yes, sir. Pisum.
Okay. Pisum sativum. Okay. Very good. What type of progeny was obtained by Mendel in F1 and F2 generation when he crossed?
In the F1 generation, the progeny was all tall, sir. And when he self-crossed them in the F2 generation, they were both tall and short in the phenotypic ratio of 3:1.
Okay. So in F1, only tall. In F2, tall and dwarf. Correct? Now tell me the ratio, GT ratio in F2 and
1:2:1.
1:2:1.
GT ratio in F2.
3:1.
1:2:1. 3:1. Correct?
Yes, sir.
Okay. What this one represents? Tell me the genotype.
Sir, it's homozygous as to heterozygous as to homozygous.
Okay. This one is genotype. Tell me the genotype.
Homozygous tall. Capital T, capital T.
Okay. 2 is?
Heterozygous tall.
Capital T and small t.
Then homozygous dwarf.
Correct?
Yes, sir.
Okay. Okay. So all of you did this question well?
Yes, sir.
Yes, sir.
Okay. Very good. Now, same kind of question, right?
Yes, sir.
Have you all done this question?
Yes, sir. I
Yes,
wrote
sir.
that all.
You were asked to do the crossing. Okay, you all
Yeah,
did
and I
this?
drew Punnett square.
Huh?
Punnett square I drew.
Okay, you did Punnett square. And what was Mendel's observation? What did you do?
Sir, he observed
you
that
just-
in one generation, all the plants were tall only. There were no dwarf plants.
Only that one. Explain Mendel's observation when he crossed a homozygous tall plant with homozygous dwarf plant by self-cross. Monohybrid cross, full.
Yes, sir. So when the plants in like the F1 generation were self-crossed, they gave rise to both tall and dwarf plants. Again, in the phenotypic ratio 3:1 and genotypic ratio 1:2:1.
Okay, so same thing you wrote, but did anybody connect this with the law of dominance, law of segregation?
No, sir. I just wrote the observations.
Okay, just you worked out and have given the thing, but maybe a kind of repeated question, right? In different aspect. Okay, fine. If you have done that monohybrid cross and given the ratios, that's better. Like this, the one which we did for the previous question. Okay. And then coming to the last question now. Next question. How many of you did this? Is this the last question?
Yes, sir. I just did Punnett square and I wrote the observations.
Yeah, sir. So did I.
You tell me now what you have done. I will do it here. You correct me. You're going to guide me. Let me check what you have done. First, what I have to do here.
4x4, you have to make a table. 4x4 table you have to draw, sir, first.
Oh, just like that I can go for 4x4 table, No.
Sir, like first, you're asking for Punnett square only, no, sir?
Yes, ma, Punnett square. But complete thing, no, for both F1 and F2 generation.
Okay, so first that I didn't do a Punnett square, sir. I just wrote it down for F1 generation.
Okay, but in the sense, you have to do all these things.
Yeah.
Like this.
Yeah, I didn't draw Punnett square for F1 generation. I wrote the observation.
Okay.
For F1 generation.
Now, let me tell you how it has to be done. Like this, you have to make. Okay? Into. Correct? This is how?
Yes, sir.
Like
then
for
I
both F1 generation. What comes here?
Next, I wrote gametes. And I
gametes.
wrote the capital
What are
R,
the gametes? What are the gametes? Gametes. How it has to be written?
And
Again?
on the other side, it's small r, small y.
So, together it is?
Capital R, capital Y, small y.
Correct?
And there next to that, I wrote...
here what you have to write? Listen, here you have to write it is F1 progeny or F1 generation. Okay? F1 generation or F1 progeny. See, this carries equal importance. You have to mention. So, cross between this and this, you have to do this first. Okay? And then only you can do kind of selfing or like this selfing. Okay, now list the gametes, how the gametes will form. How the gametes will form.
Capital R, capital Y.
Okay.
Capital R, small y.
Okay.
Small r, capital Y.
Okay.
Small r, small y.
Correct? All of you got the same combination? All of you got it?
Yes, sir.
All of you did the same thing. You must have done this first. Okay, first you have to do all these things. Okay, fine. Next, what you did? I will work out the rest. Let me work out here the rest part. Okay, so Punnett square. Correct?
Yes,
for F2. For F2 progeny prediction. What should I write here?
Capital R, capital Y.
Here?
Capital R, small y. Small
Here?
r, capital Y. Small r, small y. And same
Here?
you will write down also. Same.
Correct?
Yes, sir. Now we
So
just literally multiply them.
Okay, so what comes here?
Capital R, capital R, capital Y, capital Y. Yeah. Capital R, small r, capital Y, capital Y. Then capital R, small r, capital Y, small y.
Here?
Capital Y, small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. Capital R, small r, small y, small y. Capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Small r, capital Y, capital Y. Small r, capital Y, small y. Then capital R, small r, capital Y, small y. Capital R, small y, small y. Small r, capital Y, small y. Small r, small y, small y.
Correct? All of you did this?
Yes,
okay. Now, how many genotypes you got? How many genotypes you got? 9 genotypes. Okay, you must have got 9 genotypes. Okay, tell me.
Capital
First,
R, capital R, small, capital Y, capital Y. That will be only one.
How many you got?
One. Only one.
1. Next.
Capital R, capital Y, small y.
How many?
2, sir.
And in the table? Tell me the table number. I'll just a minute.
Yeah.
I'll mention the table number also for you. Okay, just for your friends. Okay, tell me now.
5 and number 2.
Okay.
Yes, sir.
And then
capital R, small r, capital Y, capital Y. 3, 9,
just a minute.
uh, 9.
Just a minute. Just a minute.
Yes, sir.
Okay. 3 and then
9.
9. Okay.
Yes, sir.
How many total? 2.
Yeah. Then capital R, small r, capital Y, small y.
How many?
4, 7, 10, 13.
So total
4.
4. Okay. Next.
Capital R, small y, small y.
Box number
6.
how many?
Only one, sir.
Next.
Capital R, small y, small y.
Table.
That will be 2, 8 and 14.
Okay. Next.
Small r. So rest all are 1, 1, 1.
Really?
Yes. No, except like 12 and 15 and that's it. Capital Y, small r,
How many?
small y,
This
small y.
genotype how many?
Only one, sir.
1.
Then small r, capital Y, small y, that is 2, 12 and 15. Then small r, small y will be 1.
Okay, fine. So this is called what? 1:2:2:4:1.
Genotypic ratio of F2 generation.
We call it as what? They are called genotypic ratio. Okay? They are called what? Genotypic ratio. Tell me the genotypic ratio.
1:2:4:1:2:1:1.
All. Okay, that's the genotypic ratio of F2. Okay?
Yes, sir.
Phenotypic ratio?
9:3:1.
9:3:3:1. What is 9?
Sir,
Round yellow.
round and yellow.
Tell me the phenotype then.
For each one, sir?
Yeah. Round.
And yellow. Second
Yellow.
one is also round and yellow.
Round. Yellow.
Third one is also round and yellow.
Round. Yellow.
Fourth is also round and yellow.
Round. Yellow. Together
Fifth
how
one
many?
is round but green.
How many?
That's
Together.
9, sir. 9.
9. Okay, 9 are round yellow. Okay, fine. This one?
Then 5 and 6 are round and green.
Round. Green.
That's
okay.
3.
Round. Green. So together it's
3.
3.
Then we
And
have
here?
wrinkled and yellow, 7 and 8.
Okay. How many wrinkled
That is
yellow?
again 3. 3.
Okay.
And last one is wrinkled and green, which is only 1.
Just 1. So 9:3:3:1. Correct? You have written it here. The phenotypic ratio.
Yes, sir.
Right? Okay. Wonderful. You answered this one. Now, you know the combinations. Like you worked out this. And you know the combinations also produced in F2. These are all the combinations. Like, what are the combinations? You highlighted this one. Progeny where? Round yellow, wrinkled green, wrinkled yellow, wrinkled green. Round yellow, round green, wrinkled yellow, wrinkled green. Okay, fine. Now, this is a very important question. Give the reason for appearance of new progeny. What
So
is new
dominant
progeny?
and recessive characters are present. So the appearance is based on the dominant gene if present. If two recessive ones are present, then it will show the recessive character.
Okay, what else?
The traits are acquired independently.
Yeah. Did you state the law?
Yes, a law of independent assortment.
Yes, sir. I also did.
Yes, that's very, very important. Give the reason for appearance. Like, you must state the law, because inheritance of two genes from the same character, okay, alleles from the same parent, are not independent of each other. That's why there is shuffling. Like, the parental characters are, you know, round and yellow, wrinkled green. They are the parental characters. You have round yellow also, wrinkled green also. Okay, you have round yellow and wrinkled. Both are the top one. 9:1. They are the parental character. And these things are what? New progeny. Okay, new progeny means what? Appearance of new progeny. What are the new progeny appeared?
Round green and wrinkled yellow.
Yeah, round green, wrinkled yellow, shuffling of the parental characters. That's a new progeny. That is independent assortment. Like, genes can means independently assort. Okay, so that is very, very important. You would have mentioned that also. Okay, fine. So all of you did the same thing. You all got? Hello, students?
Yes, sir.
Okay,
Yes, sir.
let me check your paper, how you have done that. Fine. Next. One more question, right?
37.
This one?
Yes, sir.
Yeah. And this is a very common question, but still. Tell me what this question is about.
Enzymes. It's about enzymes. And the preparation of ethanol.
Okay, fine. So, what is the role of enzyme in general?
It acts as a catalyst when complex substances are broken down into simpler substances.
Okay, fine, good. Okay, so what is the first question? And what did you answer?
Sir, I gave the
A
answer.
statement of what
The first line of the paragraph is the answer, sir.
First line of the paragraph. Like that, you splitted all the sentences into different answers, huh? First
No,
sentence.
sir, but I derived the answer from the first line.
Okay, just cut, copy, paste, huh?
No,
What
sir.
do you understand by fermentation? The breakdown of complex substances into simple substances by the action of enzymes released by anaerobic bacteria. It's called fermentation. Okay, good. Accepted. Correct only. And then, what is molasses? What did you write for this?
Sir, I
third line, sir. Sugarcane juice
Yes,
and
sir.
water are mixed and heated to form a sweet liquid called molasses.
The same sentence, you cut, copy, paste. Did all of you did the same thing?
Words I
slightly changed.
Yeah, it's the first 3 words I cut it out.
Oh, first 3. Okay, very good. Wise job. Very good. sugarcane juice and water are mixed and heated to form a sweet liquid called molasses. Right?
Yes, sir.
Very good.
Somewhat, yes,
Okay.
sir.
Okay. Well, what is the third question? Which method is used for the preparation of ethanol?
Fermentation.
Fermentation.
Huh?
Fermentation, sir.
Or you did the cut copy like this. This method is generally carried out in breweries. How many of you did this?
No, sir, it's actually in the second line,
No,
sir.
sir.
No.
But I didn't do it, sir.
Okay, this is the correct one, right? This method is generally carried out in breweries. Okay, fine. So what did you write?
Fermentation.
method?
Fermentation, sir.
Fermentation? Only fermentation?
Yes, sir.
Okay, only fermentation. But what is the correct one? It is, in fermentation, you get different product, right? Your idli dosa batter is again fermentation. Your bread making is again fermentation. Now, this is about ethanol. Ethanol is not just by fermentation. It is specifically what?
Fermentation of sugarcane juice.
No. What method is used for preparation? It is anaerobic
Anaerobic
fermentation.
respiration.
Okay, that anaerobic word is very, very important. Key. Important. Okay, key word. Okay.
Okay, sir.
Anaerobic fermentation. It is anaerobic fermentation. Okay, fine. Then, what is the general mechanism by which enzymes acts? Very, very important question. What is the general mechanism by which enzyme act?
When they catalyze the chemical reaction and they dissociate the product.
So listen, look into the image. Okay, how enzyme acts? See, based on the picture.
Attracts the substrate.
Listen, enzyme binds with substrate. Okay?
Yes,
Gives,
sir.
which forms what? Enzyme binds with
Broken
the
down
substrate and forms enzyme substrate complex. And then it forms enzyme product complex. And then it forms, the enzyme comes out as such. See, look at this fellow. 2 eyes. 2 eyes. Can say 2 air. 2 air. And one open mouth. One open mouth. What it represents? Any change in his look,
The
appearance?
catalyst is not involved in the reaction. At the end, comes back.
Look, in the sense, see, this fellow do not change its structure or whatever. In the initial stage as well as in the end stage, the structure of the enzyme remains as such. Correct? This enzyme did not go any conformational change. Okay? But look at the substrate. On green fellow become of green of pink. Means the product. The substrate is converted into product. Okay? So enzyme plus product. Okay? The direct answer for the question is this one. Enzyme. This is the question like, what is the general mechanism by which enzyme acts? Enzyme binds with the substrate. It forms enzyme substrate complex, and then it forms enzyme product complex, and finally enzyme comes off the reaction by producing the product. This is the simple direct answer. And there is one technical and indirect answer also, which is good for you to know. I will tell you. See, something is called what? The main action of enzyme is called what? It increases the first one. You can list the characters of enzymes. Okay, enzyme action. Enzymes, what it do? It increases rate of reaction. What is called rate of reaction? The speed of the reaction. That's why they are called biocatalysts. And then decreases activation energy. Okay, decreases activation energy. What is called activation energy? See, this is a thick iron rod. Okay, this is thick rod. To break this rod, okay, this is the substrate you can say. To break this rod, what the enzyme will do, it should come and bind and it should bend it. Okay, the enzyme should come and bind like this and it should bend it. So, the rod should be first, imagine you're breaking a stick like that. So, the substrate should bend first. This structure is called what? Transition state. What is this state? The substrate is about to break. Okay, imagine you're bending a stick. That is the function of enzyme. Okay, in the sense, to break a rod, you need more energy. Activation energy means the energy required for bringing the substrate to transition state. Substrate or the original state is this. Transition state is this. To bring the substrate to transition state, you require energy. That energy is called what? Activation energy. Okay. So, enzyme increases rate of reaction. Immediately it does, and then it decreases activation energy. So, easily it breaks the substances. Bring the substrate into transition state. Thus, it breaks the product. Okay. It increases rate of reaction, decreases activation energy, converts substrate to product. Okay, so this is the technical answer. Since you are in class 9, this explanation is marvelous. But when you go to your higher studies and all, in 10th, the same chapter is there, how an enzyme acts. There, you have to talk about all these things. Enzyme increases rate of reaction. Enzyme decreases activation energy. Enzyme converts substrate into product. Okay, so these are all the 3 important points you have to give for this particular question. Okay, is it clear? Hello, students?
Yes, sir, it's clear, sir.
Fine. So, and when is the next?
How exactly do we define it?
What?
So how should we write it like in a single sentence? How do we define it?
For this question, now, here,
Yes, sir.
here if you write this enzyme binds with the substrate, look at the image based on this enzyme binds with the substrate, the enzyme binds with the substrate, makes enzyme substrate complex, and then it makes enzyme product complex, and finally enzyme, and then it makes the convert the substrate into product. Enzyme comes out of the reaction without any change. It brings the substrates into product. Okay, if you write this in the sentence form, how enzyme work? Diagram is given, you have to give the explanation. That's the question. General mechanism by which enzyme acts. How enzyme work? This in sentence form. Enzyme binds with substrate, forms enzyme substrate complex, and then it forms enzyme product complex, and then enzyme comes out of the reaction by releasing the product. This is the general mechanism. Okay, this only you have to answer. That's the question is asked. Okay?
Yes, sir.
Yes. So you enjoyed writing the test? Hello? Students?
Yes, sir.
Okay, good. So, any more doubt in this particular chapter?
No, sir.
Okay. Heredity and evolution. So no questions from evolution side, but heredity. So be thorough with in this, like focus much on the terms connected with heredity and evolution. Okay, what are the different terms? Genes, alleles, multiple alleles, homozygous condition, heterozygous condition, genotype, phenotype, dominant gene, recessive gene. Okay, genotypic ratio, phenotypic ratio, F1 progeny, crossing, self-fertilization, cross-fertilization. Okay, so many terms, law of dominance, law of independent assortment, law of segregation. Okay, so these terms alone, very important, will help you to understand most of the concepts. Okay, the terms are very important, right? Be thorough with the terms. Okay,
Okay, sir.
yeah. So what's the next test? What chapter it is? When is your next test? In biology, what is the chapter?
I think it's how do organisms reproduce, sir.
Okay, how do organisms? Hope you all prepared well for the test. Okay, I think are we having any revision class for that? Thursday, do we have any class? Thursday or Saturday? Any class? It's planned? Anyway, let me check and tell you. But be prepared. Okay?
Yes, sir.
Yeah. Thank you. Thank you so much. Take care.
Thank you, sir.
Yeah. Good.